Okay, this video, like the organic compound video, should be mostly all review from your freshman year. There'll be a little bit in the end that maybe is new to you. Okay, so we're talking about enzymes here. And if y'all remember, enzymes have an impact on our activation energy of a reaction. So the activation energy is going to be the energy that is required to start the chemical reaction. It's the energy that we need to break the bonds in the reactants. So you can see on your little graph here, you've got your reactants. It's the energy we need to break these bonds, okay, so that they can be reformed into these new products. So our activation energy would be this part right here. Okay, you can see how these bonds here get into this transition state, so they're able to be broken, and now we can reform them into products. So that's the activation energy, that energy required to start the chemical reaction. And your catalysts are going to be anything that decrease activation energy without being consumed. And you've got inorganic catalysts as well as organic catalysts. And we're going to focus on organic catalysts, which are our enzymes. Okay, so they're kind of organic catalysts. So they're going to lower this activation energy without being consumed. So let's take a look at some characteristics that are enzyme specific. Okay, so enzymes are going to be made of proteins. So their monomer, would be an amino acid. They're made of proteins. We do have some enzymes that are made of pieces of RNA, but that is a relatively recent discovery. So our discussion will be focused on the protein aspect. So most of our enzymes are going to be made of proteins. So they would have an amino acid monomer. If you can look at this enzyme over here, okay, we can see this is our protein chain. We've got our green, okay, and there's our alpha helix, our beta pleated sheet. Okay, that protein has folded on itself with its tertiary structure. So its function, remember, is going to be based on its structure here. Okay, enzymes are also substrate specific. So if I look down here at this one, okay, right I have down here is what's called ribonuclease. Okay, and if you remember, enzymes end in ASE. Okay, so I have ribonuclease. There's my ASE for my enzyme. Okay, so my enzyme is the purple part here. That's my enzyme. Okay, and my RNA here is this green part. Okay, and so what this enzyme is doing is it is breaking down RNA so that those nucleotides can be reused. Okay, so ribonuclease. Okay, enzymes are also, as we mentioned on the first slide, enzymes are not consumed. Okay, they are reusable. Okay, and so they are not consumed during the chemical reaction. The fourth characteristic is what's called induced fit. Okay, so you can see here in this top picture, okay, we've got the active site for the enzyme. Remember, the active site is where the substrate binds. So remember, the active site is part of the enzyme. Okay, so that's where the substrate is going to bind. And the substrate, so here's my active site. The substrate is the molecule that is binding with the enzyme. Down here, see, I've got these substrates. Okay? They're the molecules that are going to come into the enzyme, that the enzyme is going to act upon. Okay? And as we mentioned on the slide before, enzymes are substrate specific. And they're substrate specific loosely based on shape. Okay? You notice the enzyme of this active site here is kind of indented. Okay? And this substrate molecule can fit in there. And then you'll see the enzyme kind of changed its conformation. Okay? It kind of closed off that active site and changed its conformation of shape a little bit. Okay? That's what we're calling this induced fit here. Okay? That the substrate may not fit perfectly into the enzyme, and the enzyme will alter its shape just a little bit to really make the chemical reaction happen, to make the substrate fit well. You can see that down here at the bottom. Okay? Here's my active sites marked by these stars, these asterisks down here at the bottom. Okay? This active site is loosely the shape of this first substrate that's headed in there. Okay, as that substrate heads in there, the enzyme changes its conformation to fit around the substrate better. So now I get this nice tight fit around my enzyme with my enzyme substrate complex. Okay, so now that we've looked at some characteristics of enzymes, let's just kind of walk through an enzyme happening here, an enzyme reaction happening here. So I've got my substrate, that's my molecule that's going to bind, my sucrose, it's just a disaccharide, okay, just a two monosaccharides bonded together. So they are going to bind to the active site of my enzyme. So they bind to the active site of my enzyme. I get that induced fit, slight change in the conformation. Water comes in, and you see my products are two separate pieces. Here, I've got 1 glucose and 1 fructose. So what kind of reaction was this? Well, that would be a hydrolysis reaction. And then now my enzyme is ready to be used again. And my enzyme is not consumed. Okay? And so my enzyme, the benefit to my enzyme, again, like we mentioned in the very beginning, is that it can lower the activation energy. So if we look here, okay, we can see how that's done. Okay, so if we look at this graph here, I've got my original activation energy without the enzyme. Okay, it's the black line there. It's very high. Okay, and once I add that enzyme, okay, so it would require all this energy right here to be able to break these bonds here between that fructose and the glucose. Okay, and when I add my enzyme, that activation energy drops significantly. Requires much less energy to make this chemical reaction happen and to form the products. Okay, if the analogy we used your freshman year would be, if you have to run a marathon and to get to the starting line of your marathon, you have to run over a mountain or you have to run over a hill. Well, most of us would rather run over a hill before we start our marathon. Okay, so the energy to get to the starting line of the marathon, okay, would just be this, as opposed to all of this. Okay, so if we only run over a hill to start our marathon, then our reaction is going to happen a whole lot faster. So let's look at some rates of the chemical reaction. That's what you guys are going to be focusing a lot when you're doing your lab later this week. Okay, so on this particular graph right here, okay, I've got my amount of product that's being formed here on my x-axis. Okay, I've got my time period. Okay, and I've got a fixed amount of enzyme and a fixed and excess substrate, way more substrate than I need. Okay, and so what I have here is my initial velocity is always going to be faster. My velocity here is going to be the slope of my line. And my initial velocity is always going to be faster because my reaction is going to start off faster. I've got plenty of substrate at the beginning. I haven't started breaking anything down, okay, or building anything up. I've got plenty of substrate available for the enzyme to work with. So initially, my reaction is always going to happen faster based on simple kinetics, right? The more substrate I have available, the more likely it is to essentially to bump in or run into the enzyme. So now let's look at if I change the substrate concentration. On the last graph, there was no change in the substrate concentration, a fixed amount of substrate concentration. In both of these, as I move to the right, my substrate concentration is increasing. Okay, and so on this top one here, you can see I've used 11 different tubes. And right when I hit about tube 7, things started kind of evening out in terms of my initial velocity of my reaction. Okay, well, what tube 7 would be, right here on this graph, would be point B. Okay, and that would be what's called my saturation point. Okay, and so essentially when I've reached there, okay, my enzymes cannot work any faster. You're basically at the point where the enzyme, okay, is working as fast as it can. There is enough enzyme to work with every single substrate molecule that is available. Well, back that up. Every enzyme is busy. There may be extra substrate. There may be a line to use the enzymes by the substrate molecules, but they still cannot go any faster. And so that is our point of saturation. Every enzyme is busy. Every enzyme is doing the chemical reaction that it's trying to do. And so it just can't go any faster. So it evens out into this straight line. Doesn't matter if I keep adding more and more substrate. And you can see here when I get to tube 11, okay, I've been adding more and more substrate, and that doesn't matter. My reaction is still going to eventually level off. So let's look here on this slide. We're going to look at two things that can affect the rate of the enzyme, of the chemical reaction here. So the first one we have up top here is temperature. So you'll notice as I move into the right here, I'm increasing my temperature. And most enzymes have an optimal temperature. Okay, you can see here the first one, the black graph there, is your optimal temperature for your typical human enzymes. Okay, and then over here, my red is my optimal temperature for my thermophilic or those very heat-tolerant bacteria. As a general rule, okay, as I increase my temperature, the rate of the reaction will increase to a point. Okay, due to, again, based back to kinetics. Okay, as I increase temperature, the molecules move faster, so they are more likely to collide with one another. However, the problem we have with proteins, okay, is that eventually the heat becomes too much, and the heat breaks apart those hydrogen bonds. And that's what we have happening here, where they very quickly, okay, that reaction plummets, okay, and it's because the heat is too much, and the heat is breaking apart those hydrogen bonds, and so that protein starts to lose its secondary structure, And remember, when a protein loses its structure, it can't do its job. Down here at the bottom, we have pH. pH can also do the same thing. Okay? Most human enzymes have an optimal pH of around between 6 and 8. Okay, with the exception here that we have here is pepsin. Its optimal pH is down at about 2, and that's one of the enzymes that's found in your stomach. But pH can have the same effect as temperature. Too alkaline, too acidic, you should have these relatively quick drop-offs. Okay, that's because, again, you get to that denaturation. You alter that pH enough, and it can denature the protein, disrupt those hydrogen bonds again. And if it disrupts those hydrogen bonds at the active site, then that protein will stop working. Okay, so let's look at inhibitors then. I've got two types of inhibitors here. I've got a competitive inhibitor versus a non-competitive inhibitor. So my competitive inhibitor is going to compete with my substrate, and it's going to compete directly for that active site. Okay, so the competitive inhibitor would slow down my reaction rate because sometimes that enzyme is going to bind with the competitive inhibitor as opposed to the substrate. You'll notice if you look at their shape, they're about the same shape, so that competitive inhibitor can fit into that active site. Our non-competitive inhibitor is also going to decrease the reaction rate. Okay, so both of these things are going to decrease reaction rate. But you'll notice my non-competitive inhibitor does not directly compete for the active site. Okay, the active site is still free, but the non-competitive inhibitor binds somewhere other than the active site, so it's bound over here to somewhere other than the active site. But when it does that, it changed the shape of the active site. So now that substrate cannot fit into the active site anymore. So that would be our non-competitive inhibitor. So when we're talking competitive and non-competitive, we're talking about whether they're competing directly or indirectly for the active site. So the next thing we have here is what's called allosteric regulation. Okay, and so allosteric regulation, there it is right there. Okay, allosteric regulation only involves proteins or enzymes that are going to have at least 2 polypeptides. Remember we talked about some proteins have that quaternary structure because there's more than one polypeptide that's interacting to form the protein. So these are going to be, allosteric regulation is going to occur on enzymes that have 2 or more polypeptide chains, and each one of these polypeptide chains have their own active site. So if you can look here, we've got these four purple polypeptides here, okay, and they each have their own active site. Okay, so every single one of them has an active site. And so what happens here is the enzyme has 2 conformational changes. It's got an active form and an inactive form. Okay, and it can bounce back and forth between the two, which are essentially a functional form and a non-functional form. Okay, and what will happen is that there's going to be a place where what's called an activator can bind. That's your allosteric site. Okay, so there's a place where an activator can bind to this enzyme. And when the activator binds to the enzyme, it will stabilize it into this active formation. Okay, and you can also have an inhibitor that would bind to that allosteric site, which would then stabilize it in its inactive or non-functional form. So allosteric regulation can either contain activators or inhibitors. Okay, they're going to bind not to the active site. You see that there. They're binding to the allosteric site. But what they do is they stabilize the conformation of the enzyme. Okay, they stabilize its shape in either a functional form or a non-functional form. Feedback can also influence the enzyme's ability to function. As a general, this can be negative feedback. Okay, and so what we have here is most of our enzymes are parts of a pathway. The example we have here is my initial substrate being threonine. Okay, threonine binds to enzyme number 1, okay, and my end product is going to be isoleucine. Just notice I had this long chain of enzymes. It had to go through a long pathway to get there. Okay, enzymes are rarely solo. And so what happens is my product, okay, so my isoleucine here, my product is going to circle back and it's going to shut off this first enzyme. Okay, you can see here there's an allosteric site on that first enzyme. You can see there it's empty. And when that isoleucine binds to that when there's plenty of isoleucine available, it will bind to that allosteric site, okay, and it's going to cause that enzyme to become non-functional. It acts as a non-competitive inhibitor. Okay, and so then when there's not enough isoleucine available, it will release, and that enzyme will be functional. Okay, so the last thing we need to talk about is cooperativity. Okay, let me spell that one out for you guys. Okay, so cooperativity is always a good thing. Again, this is like similar to allosteric regulation. I've got enzymes with two or more subunits, two or more polypeptide chains, and they each have their own active site. Okay, and here's my inactive form over on this side. Okay, and so with cooperativity, I've got, when I can get one substrate to bind to this enzyme, that's going to conformationally change all the rest. Okay, and so that will make all the rest of those active sites be more easily accessible. Okay, and so cooperativity is always going to be good. Okay, so I've got my inactive enzyme. All I need is just one substrate to bind to it, and that'll conformationally change the rest so that they can easily bind to their substrates.